Hey, happy Friday, folks. We're going to pick up right where we left off today, right back into membranes. If you remember, we were talking about membrane structure, and today we're going to talk a little bit about membrane transport, and we might get to the section on cell walls. So I had left you with this kind of question, which is kind of the basis of a midterm question that I often give that's asking you to describe the fluid mosaic model. I might not be quite so open-ended. I usually try to be a little bit more specific around what I'm looking for. But just a reminder, what the fluid mosaic model is it really is talking about the phospholipids and proteins. So make sure that you do put those in there. And I even drew a little picture last day. I drew another picture today. I'll show you in a moment. Your answer may include other parts of a membrane, and those type of things aren't going to hurt, but they're not necessary for this kind of question. So fluid mosaic model, right? The fluid part, of course, is the phospholipids. So you're gonna, if I'm asking you to draw a diagram, you're gonna draw some phospholipids. Maybe they don't have to be this fancy. I know some people are really artsy and some people are just barely getting by. So that's me, just barely getting by with the artsy stuff, but you can do it. You can draw a little circle with some little tails on it. You can see I have it labeled, polar phosphate head, and you've also got the non-polar lipid tails. It's very important to use the language of the course, so I would expect that in your answer you talk about there being a hydrophilic or polar component to the phospholipid bilayer, and also very importantly the hydrophobic zone as well. The mosaic part, of course, oh, there we go. There's the mosaic part. That's going to include the membrane proteins. So you're going to have membrane, integral membrane proteins. Maybe you'll have some peripheral membrane proteins, and you're going to talk about how, in this zone here, right, there's going to be hydrophobic amino acids. And this zone here is going to be full of polar amino acids because it's interacting with the water. So you can see I've added some cholesterol. That's not necessary for the fluid mosaic model, but it is kind of just there for completeness. I've thrown in a couple other things there. You can see I've got some glycoproteins and some glycolipids, and that's kind of the model of the membrane. It is a little bit more complicated than that, but this is kind of the basis and really what we're covering in Biology 107. We do need to move on, and I want to talk about membrane transport. So today we're talking about membrane transport, and you've got this phospholipid bilayer, and it's got these chemical properties to it. It has a hydrophilic zone and a hydrophobic zone. So if you think about what can easily get across a membrane, really it's going to be things that can get across that hydrophobic zone. So what does that mean? That means really small molecules. So by small molecules, I really mean three things. I mean water. Water is very polar, but it's very, very small and found in great abundance in a cell. So when you have something in great abundance, some of it's going to get through. So water gets through for those two reasons, small and very abundant. The other small molecules that we're really referring to are oxygen and carbon dioxide. And oxygen and carbon dioxide, of course, are important gases that we bring in oxygen and we exhale carbon dioxide, of course. Both of those happen to be hydrophobic molecules as well, which is the third group here. So hydrophobic molecules can get across the membrane relatively easily. So you're probably wondering, hydrophobic molecules, are they even going to be found in solution, like in your blood or in your cells? Yeah, in small quantities. Hydrophobic, it doesn't mean they're completely insoluble, but it just means you can't get a lot in solution, just very small amounts. And sometimes that's the case with a lot of drugs or lipids. They can be found in very small amounts in solution, and they can get across the membrane very, very easily. So what does not pass easily? Pretty much everything else. So that means large things, it means polar things, and I guess you can throw charged onto that group. Polar, charged, I kind of think of them as very similar chemically, at least from a membrane perspective. So you've probably talked about various types of transport before in high school, I'm guessing. So hopefully this will be some review for you. I'm going to just kind of go through this relatively quickly. If you need me to slow down, just let me know. The first type of transport is diffusion or passive transport. And what does this mean? It means you have molecules that are moving from an area of high concentration to an area of low concentration. So they're going with the concentration gradient, and there's no energy required. That's why it's called passive. So I've already mentioned oxygen and carbon dioxide fall into this category. They can just pass across a membrane relatively easily, which is good because it means we can get oxygen to our tissues and we can get rid of that carbon dioxide relatively easily without any special transporters. There is a special type of passive transport that's involved in water, and this has a special name. It's called osmosis. So I'll just play that animation for you again here, and you can watch. You'll see that the green molecules are not passing the membrane, but the water is passing the membrane relatively easily. So you can see there it goes. There goes all that water back and forth. So this is osmosis. I have another video to show you, and often when we're talking about osmosis, we're thinking about what is going on with solutes, right? So if you and this will become clear when I get to, and I'm going to start to talk about animal and plant cells in a few moments. But I'll show you this other video. I can advance to the next slide. There we go. So here's a little video here. You can see it's going to play, and all those little water molecules, they cross through the membrane very easily. Now in a moment, this person's going to add some salt. And so the salt is added on the right side, and what's happening is the water is moving from the left side to the right side of the stream. So why is that? This is basic osmosis, is that the salt cannot cross the membrane, but the water can. So if you think about what's going on the left here, on the left we have, maybe 100% water. So that's high concentration. You add a little bit of salt. It doesn't even matter what concentration of salt you have. All we know is that it's less than 100%. So it could be 99%, could be 80%, it doesn't really matter. But when you have a high concentration and low concentration, and that molecule can cross the membrane, it goes across the membrane, and so the water on the right side rises. So that's osmosis. There's all sorts of experiments you can do with osmosis. Something that you can do is you can buy these things called these artificial membranes. And you can see in this experiment here, we have an artificial membrane. It has a 2% sucrose solution. It could be salt, it could be all sorts of things. Sucrose does not cross membranes very easily. And you can see I have 3 solutions: a distilled water, 10% sucrose solution, and a 2% sucrose solution. So some terms for you. And I'll kind of give you some hints about how to remember what these terms are: hypotonic, hypertonic, and isotonic. So tonic means, solutes or something like that. Hypo means not enough. So if you are hypothermic, that means you're too cold, you don't have enough heat. Hyper means too much. So if a child is hyperactive, it means they have too much energy. So take a look at what happens here, and I'll show you what happens with certain types of cells in a moment. But in the distilled water, this is a hypotonic solution, so the water is going to go in the direction of the solute. It's going to go from the beaker into the artificial cell. In the hypertonic solution, it's the exact opposite. It's going to go from the 2%, which is 98% water, to the 90%. It's going to leave the cell. And then the isotonic water is going to go move in and out at the same, roughly at the same rates. So this has implications if you're an animal cell in particular, because animal cells really like to be at isotonic solutions. So what you're looking at here is, this is a little cartoon of some red blood cells. And take a look in the middle diagram first. You can see water is going in and water is going out at the same rate. And in that case, the cell is very, very happy. If you look on the left, it's a hypotonic solution. So that's maybe 100% water. And water is mostly moving in. And so the cell is growing and getting fat. And for an animal cell, that's a bad thing because the cell will probably burst open. So by the way, this is how I remember which is a hypotonic, by the way. Hypo is kind of like the word hippo, hippopotamus. So you can see that the cell is getting big and round like a hippopotamus. So that's another way that I remember which is which. You can see on the far right, the exact opposite is happening. It's a salty solution, so the water is departing the cell. So if you're an animal cell, you really want to be isotonic because that keeps your cell in good shape. This is not necessarily the case if you're a plant cell. So you probably know if somebody gets you flowers, what do? Well, you put them in water because you want to keep them nice and fresh, and it keeps them from wilting. So for plant cells, isotonic solutions aren't necessarily the best. They may get a little bit wilty in an isotonic solution. What you want to do is give them lots of water. And so in that case, the water runs into the cells, and they have a cell wall, and the cell wall prevents them from rupturing. In the end, the cells end up nice and turgid. If you think of turgid as like a nice crisp carrot or vegetable, it's in good shape. And this is how plants keep themselves in good shape rather than wilting and falling all over the place. They want to have lots of water. The opposite of that is plasmolyzed. You can see on the right there, that cell is getting plasmolyzed, which means it's in salt water. And so the water is departing the cell, and you can see that the cell membrane is actually kind of tearing away from the cell wall. I have a little video showing you some cells getting plasmolyzed. I think that's, maybe it's on the next slide. What I want to show you is an animal-like cell here that is actually doing okay in pond water. So there's all sorts of organisms like this. This is a paramecium, and a paramecium is, it's a protist, but it's animal-like in that it doesn't have a cell wall. And how does it survive in pond water without turning, bursting open like a hippo? It actually has something called a contractile vacuole. So you can see it's listed here and shown right there. And that's what I have on that arrow. So if you watch this video carefully, every once in a while, it takes about maybe 10 seconds. I'm just watching it now. And you'll see eventually a circle, and it's going to pump the water out. So that's what a contractile vacuole is. There it goes. Contractile vacuole. It basically pumps water out. And this is an organism we normally look at in the biology lab here. Unfortunately, you won't get a chance to do that this semester. But they're very cool to look at under a microscope. And if you watch carefully, you can see those contractile vacuoles. So a little bit more on paramecium later. So here's an organism that we were looking at in this week's lab. So this is Elodea. So Elodea is an aquatic plant. People used to have them in their aquariums all the time. You can't get Elodea so easily now because it turns out it's an invasive species, but it's actually a very nice plant to have in an aquarium. They grow well, they don't die, they look pretty. And the nice thing about them for microscopy is they have a very thin layer of cells in their leaf. So you can look at them, and it's usually 1 or 2 layers of cells, and you can make out the details there. So if you take a look, this is one cell. I'm going to just circle it. That's one whole cell like that. So I know there were at least 1 or 2 people with questions about which the cells were. These little things, those are the chloroplasts right there. And so what you're looking at, if you take a look at that picture above, you've got these cells, and you've got these little chloroplasts all around, and in the middle, you have this giant vacuole. So the chloroplast is sort of pushed to the outside of the cell because the vacuole is full of water. So normally these plants have water in them, and it's keeping them nice and turgid. So what happens if we put this into a salt solution? Those vacuoles, the water runs out and they shrink, and the chloroplasts kind of clump in the middle. So I'm going to actually show you, this is a video, and this one here on the left is a video. I'm going to press play. Oh, there, press play, and you can watch what happens in real time. The salt water is added, and you can see the vacuoles are shrinking. The chloroplasts are kind of starting to clump together in the middle. And it's getting plasmolyzed, which means, like I said, the cell membrane is getting torn away from the cell wall. So there it goes. So probably nothing new for you. You probably knew put your plants in water. Don't put them in salt water. But this is why you're killing the cells in the salt water. Okay, so that's osmosis. Sometimes there are molecules that are found in a high concentration on one side of a membrane, and they want to get across, but they can't because they're too big, or maybe they're too polar. So in that case, one mechanism the cell has is something called facilitated diffusion. So the word facilitated means helping, right? To facilitate means to help. So you have a helper protein is basically what is going on here. So there's actually a couple of different types of these things for facilitated transport. One type is called a channel protein. It's kind of just like a tunnel. I have a picture of one here. This is one found in E. coli. This one here is found as a trimer, so 3 of them are kind of stapled together. And they have, you can see that little hole, that little pore in the middle. And so basically that hole is going to be the correct size for a sucrose molecule, and also it's going to be complementary in its molecular charges. So sucrose is a polar molecule, and so this is going to be a polar channel to allow sucrose in, and then E. coli can eat. So there you go. You can see the second example is called a carrier protein, or sometimes I've seen the term gated protein. It kind of opens back and forth like a little gate. I have a little a picture of one here. This is a lactose permease, also found in E. coli. You can almost imagine that it has these hinges on it. It opens up to one end, the molecule will enter. Again, you've got another polar pocket, and then it hinges and opens up on the other side. I have a little animation to show you how this works. So there's a little animation, and you can see the molecule is going in and hinges and goes out. So unfortunately, this little animation is going backwards because E. coli wants to eat lactose. It doesn't want to release lactose to the environment, but you get the idea. The protein hinges and allows the substance to go in and out. Okay, so all of this transport we've been talking about so far has been passive, and by passive I mean it is basically following a concentration gradient and does not involve any kind of energy investment. And so we want to talk about what happens if we need energy, and this is called active transport. So active obviously means energy. So if you take a look, there's a little diagram. You've got a cell. Active transport usually involves some sort of membrane protein, and that membrane protein is often going to use some sort of energy source such as ATP. So there's the ATP, it's getting consumed. The ATP is in the cytoplasm, of course, and this pumps whatever that is in. So maybe this is a hungry cell and it sees some glucose and it wants to pump in some glucose and it wants to bring in lots of it. So it's going to take energy in order to do that. So here's a little animation showing a little ATP is getting hydrolyzed, and whatever that green molecule is, it's going in. So the classic example of active transport is the sodium-potassium pump. We'll talk about that in a minute, but looks like I just want to talk about ATP for a minute first. So I mentioned ATP already way back in the last topic. We were talking about macromolecules. Or sorry, that was topic 2. And we were looking at the structure of ATP, and I had told you it is a nucleic acid. So remember nucleic acids, we have a nitrogenous base here. We have a pentose sugar, so a 5-carbon sugar, and then we have phosphate groups. So there's ATP. ATP stands for adenosine. So this whole group here, by the way, those two things together are adenosine. And then triphosphate. So tri for 3 and phosphate for phosphate. So there's a little sketch of ATP. And how does this work? So one thing I noticed when I look at this molecule, and these are the kind of things, you take your chemistry and you learn things, and sometimes you can apply it. In this case here, I'm looking at this molecule, and what do I see? I see negative charge, negative charge, negative charge, negative charge, a bunch of oxygens. Oxygens are very electronegative, and so there's an issue with the molecule. There's tons of negative charges together. And you probably know the basics of all chemistry is that negative charges like positive charges. Negative charges do not like negative charges. So in order to put that molecule together, we had to take these negative charges and somehow get them close together. So what does that mean? It means it took energy to produce this ATP molecule. That means when I break it down, I'm also going to release energy. So if you take a look down there, there's my ATP, and we can release energy by using a hydrolysis reaction. Remember, hydrolysis is adding water, and we can make ADP, so D for di, and when we release a phosphate, we get energy. a significant amount of energy. And so this is what the cell does when it needs energy. It can use ATP for this. We can also make AMP. We can release a second phosphate and even release more energy. So I just want to point out these phosphates here. Right here you can see it's the abbreviation is PI. Usually the P is circled, but not always. That just means inorganic phosphate. Inorganic meaning it doesn't have any carbon molecules to it. So if you ever see that, it just means it's one of these phosphates and it has a bunch of oxygens arranged around it in a phosphate group. So like I said, one of the classic examples of active transport is a sodium-potassium pump. This is very active in your nerve tissue where we have charges moving across the cell membranes constantly. And it is a hinged protein, so kind of like a carrier protein, except for it's going to involve ATP. So you can see the first step there, we have 3 sodiums from the inside of the cell, from the cytoplasm, are going to bind the transporter. And so the next step is ATP comes along. So there's my ATP right there. And you can see it's getting broken down into that inorganic phosphate and that little ADP. So that inorganic phosphate actually, in this case, actually attaches to the protein. This is called phosphorylation. And what that does is it adds energy to that protein. And what does that energy do? It actually causes the whole thing to hinge. So it's like you or me, we see a gate, you push on it and the gate opens. So there we go. The sodiums get released. And then the next step is there's actually pockets in there that can bind potassium. So the potassium pockets are actually separate than the sodium pockets. Potassium is a little bit bigger than sodium, and so it actually has unique pockets for that. The potassium binds, and once the potassium finishes binding, the phosphate is actually released. So it's kind of like you or me letting go of the gate, and then the gate swings shut or swings in the other direction. And you can see now the phosphates are ready to be released to the cytoplasm. And then the cycle kind of goes over again. So what's going on in our nerve cells? We're not really talking too much about them in this class, but nerves run on electricity, right? And how is that electricity generated? By having an imbalance of charges across membranes. So in this case, you can see you're bringing 3 charges out and 2 charges in. So in the end, we're going to have more of a positive on the outside of our neuron than the inside. And so the ability there to have an electrical pulse. Okay, so we have passive transport, no energy required, and following the concentration gradient. So you can see going from the high concentration up top to the lower on the bottom. And we also have active transport, which requires energy and goes often against or if it's neutral, the concentration gradient. So I want to just switch gears and talk about another type of transport that we're going to call bulk transport. So these types of transport here are usually, usually referring to 1 or maybe up to 3 molecules. Now I want to talk about how we can move huge amounts of molecules across our membranes. And we have some processes for that. So these are called exo and endocytosis. So cyte, by the way, C-Y-T, that means cell, right? So cytoplasm is the plasm within the cell. So cytosis means the process of doing something, moving something in or out of the cell. So exo means getting something out, and endo means getting something in. So exocytosis and endocytosis. So you can see in this case here, what we're doing with exocytosis and endocytosis is we're surrounding a bunch of molecules or a large object with a membrane. And then that membrane, in this case, in case of exocytosis, is fusing with the plasma membrane and releasing those molecules out of the cell. Endocytosis is kind of the opposite procedure, where the plasma membrane is going to fold and make an invaginated pocket, and molecules are going to go into that pocket, and the molecules are going to eventually that pocket gets pinched off and makes a little membrane bound compartment. So there's different types of exocytosis and endocytosis. So I want to talk about this a little bit more, but kind of generally what is going on here? Well, sometimes cells do endocytosis for feeding. Sometimes they do exocytosis for expelling waste. Or in the case of like your pancreas cells are making insulin all the time. And this is how they secrete the insulin from the pancreas into the bloodstream by exocytosis. So I want to talk about some different types of endocytosis because there's a few different types of ways to do this. And they're different in many ways on a molecular level. And they're involved in different processes. So the first type of endocytosis I want to mention is called phagocytosis. So this word here, phago or phage, means eating. So this process here is cellular eating, phagocytosis, cytosis, eating process of the cell, right? So if you take a look here, I'll show you a video or two in a moment. But the way this works is the cell makes this invaginated pocket, and these little projections here, you can see there's a name here, pseudopodia. So pseudo means fake or false, and podia means feet. So these things here are fake feet, or I kind of think of them more like arms, but I didn't name them, so here we go. So you can see they're grabbing that object. So in this case here, it looks like we have some sort of cell that's grabbing a big food particle, or maybe it's eating another cell. So maybe this is an amoeba and it's eating a paramecium or a bacterium. It's bringing it in and making that food vacuole. Sometimes we call these vacuoles vesicles or vesicles, and they're going to fuse with a lysozyme. A lysozyme is a digestive organelle. We'll talk about lysozymes in topic 6. There's a picture, an electron micrograph, an amoeba, and it's trying to bring in, it looks like a bacterium, into a food vacuole. So the bacterium is long and skinny, and it's engulfing it using its pseudopodia. So I'll show you this video here. Okay, and we're going to see some phagocytosis in action here. We'll turn the volume down, not very good sound in the background there. So you can see in this case here, it's very fast. This is sped up a little bit, but very quickly you can see the cell is moving around. It's changing the shape of its membrane and cytoplasm and bringing these paramecium into these food vacuoles. The paramecium looks like they're trying to fight for their lives, but you know what? Sorry, I don't think they're going to make it. Such as everything in nature, right? We've got tooth and claw in action there. They're getting digested, and don't worry, there's lots of paramecium out there in the wild. There's lots of videos of that on the internet if you want to check it out. Some of them are much slower, but I thought that was one of the nicer ones. So I wanted to share this with you. Calvin and Hobbes. Hey, if you're looking for some great, study breaks, Calvin and Hobbes is a great cartoon to read. I own the complete collection. They're always fun to read in the living room in good breaks. And I saw this one here. Calvin, if you don't know what Calvin and Hobbes is, it's about a little boy, and he's kind of mischievous. And there he is. You can see he's crawling along. He's got a sheet on his back, and he says, "The giant amoeba slides along the kitchen floor. He extends a cytoplasmic pseudopod," there's that word, pseudopod, "and he's grabbing a package of oatmeal cookies." So he's imaginative and he's sneaky and getting the cookies. So, hey, that's not too bad. Of course, he's noisy, and his mom sees that and she says, "Nice try, buddy. Put those cookies back." So I thought I'd share that with you. Check out Calvin and Hobbes. They're always lots of fun. So the second process of endocytosis is called pinocytosis. I don't actually know what pino means, but probably something to do with drinking or liquids or something like this. So this is where a cell is basically trying to bring in some sort of either liquid or solute. And it's also non-specific like phagocytosis. There's an image of an organism that's bringing in some solutes, and you can see there's a whole bunch of vesicles forming along the membrane of that cell. So I think I have a video here as well. So it's hard to find a really good video on this. This was one of the better ones I could find. I'll press play. Maybe I need to skip forward just a little bit. There we go. So there's the video, and you can see, I'm not even sure what kind of cell this is, but you can see it's basically forming these vesicles and kind of sucking in the liquid. Again, I don't really know why it's doing this in this particular video, but you can see the process in action, and it's kind of quick, but if you watch carefully, you can see the vesicles forming, and it's bringing in either liquid or solute. I'm not going to play the whole 11 minutes for you. I'll just leave it another few seconds here for you to enjoy. All right. One other type of endocytosis. This is called receptor-mediated endocytosis. This is kind of like if we had phagocytosis, which is cellular eating. Now we're talking about picky eating. So by picky, I mean we're really bringing in a very specific type of material here. So if you take a look at this, this little cartoon on the left, we've got a receptor, and that receptor is binding something in particular. So for example, something that is hard for us to digest is fat molecules. And so that's how we actually bring them in our intestines. We have receptors for different kinds of fats, and they will bring them in by this receptor-mediated endocytosis. This is also how we transport cholesterol around our bloodstream, because cholesterol is not very water soluble. So this is a very different process. You can see it's forming these things called coated pits. There's actually a special protein that forms that invagination and brings things in. So there's a picture of this happening in a cell. So like I said, think of this as picky eating as opposed to just general non-specific eating. I don't have a good video for you for this one. I do have kind of this little low quality animation though, and you can see that those receptors are binding that little UFO shaped molecule and then bringing them into some sort of endosome. Okay, let me see, 4:30, so a little bit more here for today. So there are all three processes: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So make sure you know those terms, okay? I know there's lots of terms to know for this course, but make sure you do know them for the midterm. It's going to be important. All right, so here is a quick test yourself question. I don't have a Kahoot for you today. I was going to put one together and I kind of just totally forgot. So I was able to pull up a question or two. So here is a question, and again, you need to be able to read the question and understand the language of the course and see what's going on. It says, which statements are false? So let's go through these one by one. So A, exocytosis and endocytosis are forms of active transport. So does it take energy? The answer is yes. In fact, great amounts of energy for exocytosis and endocytosis. Not one ATP molecule that changes the shape of your entire membrane, but we're talking about hundreds or thousands of them, depending on the size of the object you're bringing in or out. So that one is good. Facilitated transport requires energy. No, it does not. By definition, facilitated transport is a type of diffusion, which means it does not require energy. So this one is false. Facilitated transport requires channel or carrier proteins. Yes, that's what's facilitating the facilitated transport, is these channel or carrier proteins. So that one is good. And then the last one, osmosis is a form of passive transport. Yes, osmosis is. It does not require energy. So all are good except for B. I thought I had a little, oh, I guess I don't. Okay, so I do want to talk about cell walls for a few minutes today, but I do have this video I'm going to show you. I actually have a bunch of these videos. These are made by the textbook manufacturer, and the nice thing is they use diagrams that kind of look like the textbook diagrams as well. They're all about 3 minutes long. There's a little bit of, you'll listen carefully, you'll notice there's a little bit of humor in there as well. And I will have links to these in the PowerPoints. I can't share the actual videos because of copyright reasons, but I can usually find them on YouTube. There's usually somebody who's put them up there, and I'll share the YouTube link with you, and you get a chance to look at the video later. So I'll play this for you right now, and I just got to make sure I'm going to have sound. So how do I do this? I click a button here to make sure we have sound. Okay, so hopefully this will work. Here goes.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so hopefully you enjoyed the video. Like I said, you can check it out on the link on the PowerPoint. I think I have that posted now, so you can check that out. So a couple other things about membranes. We're going to be talking about membranes kind of directly and indirectly over the next few units. They're very important in eukaryotic cells. We have all these organelles that are basically made of membranes. So we're going to talk about that. Many of these organelles are involved in a system that we call the endomembrane system. So that means membranes inside the cell. So more on those later. We're going to talk a little bit about how things are shuffled around. So we have these proteins, they're made in the endoplasmic reticulum, they end up in vesicles, they go through the Golgi, some of them get secreted, some of them end up in different parts of the cell. And we're going to talk about how membranes are important in energy production in mitochondria. So if you take a look at this mitochondrion here, the inside has these inner membranes and they're all over the place. They're very, very important for energy production. So here's our old fluid mosaic model. Modern models kind of have a lot of more updated components to it in terms of, some of the proteins are found on the inside, some of the proteins are found on the outside, and just sort of added details and complexities to these to this model. Don't worry too much about the complex model. Like I said, this is the kind of thing you're going to learn all about over the next few years if you take more biology courses. So there's our updated model. We are going to talk about these other things. We can see on the bottom, I have the cytoskeleton, that's topic 7. On the outside of the cell, we have an extracellular matrix. We're going to talk about that next day, and also cell walls. So I'm just looking at the time here and realizing that there's probably not enough time to get into cell walls. And hey, it's Friday afternoon. No harm in letting you guys off a little early to enjoy the weekend. I know it's been a long week. It's been a long week for me. So I imagine it has probably been a long week for some of you. So I'm going to finish there. I'm just going to say one or two things about the lab reports. I've had some questions about the lab reports, and if you have more questions, please send them to me. I'm going to try to answer a few of them next week, kind of just at the beginning of every lecture. I'm going to start talking a little bit about lab reports and kind of give you some tips. I know there's some questions in particular about people wondering, you know, what exactly is due? That last 2 pages of Lab 2, there is a marking scheme, right? And so it has some questions, and some of them are Lab 3 pre-lab questions. Those are the questions you are responsible for. So open up a Word document, answer those questions 1, 2, 3, and so on, and that is what is due for the short report. If you have any questions and you need clarification, please let me know. Other than that, I hope you guys have a great weekend, and I will see you all next week. So take care and take it easy.